In this part, I'll discuss the two types of synapses, electrical and chemical. They occur throughout the body. Here we see an example of an electric synapse, and here an example of a chemical synapse. There are some differences, which I will discuss in this part. So let's dive into the details. I always have the notes here for you. The chemical synapse on the left, and the electrical synapse on the right. Now, in the previous part, I have discussed the details concerning action potentials and graded potentials, so I will leave them out here. Please remember, we need an entry of calcium ions into the presynaptic neuron in order to trigger the fusion of the vesicles containing the neurotransmitter with the presynaptic membrane and subsequent release of the neurotransmitter. The neurotransmitter in chemical synapses may be either excitatory or inhibitory, a highly selective neurotransmitter. This is a one-way transmission. The released neurotransmitter binds to either ionotropic or metabotropic receptors, so we may have on the postsynaptic membrane either ionotropic or metabotropic receptors, or both of them. This is somewhat slow, but only if we compare the speed to the speed of transmission in electrical synapses. On average, the delay is about 2 milliseconds. Now here on the right, we have electrical synapse. Presynaptic neuron, postsynaptic neuron. The cleft between the two membranes is 2 to 4 nanometers, as compared to 20 to 30 nanometers in chemical synapses. The distance here needs to be overcome by diffusion. Now here we have gap junctions. These are the electrical synapses. Both membranes are connected by the gap junctions. You see how well they are aligned. The passage here is not one-way, but two-way transmission. Ions and small molecules can pass through, like second messenger as small molecules. No neurotransmitter, hence non-selective. The transmission is always excitatory as opposed to either excitatory or inhibitory in chemical synapses. The effect is instantaneous. This is required physiologically to synchronize large numbers of cells. This is also a low resistance transmission. Now let's take a look at chemical synapses. Just to recap, we have here a presynaptic neuron, a postsynaptic neuron. At the end here, we have the neurotransmitter released, and the signal is transmitted from the presynaptic neuron to the postsynaptic neuron. Remember here, the neurotransmitter may be either excitatory or inhibitory. It is released from the vesicles, and it binds to the postsynaptic membranes, to specific receptors. Now, the effect of the neurotransmitter may be either excitatory, which we discussed in the previous part in details. Hence, the released neurotransmitter, if excitatory, will form so-called EPSPs, excitatory postsynaptic potentials. See, we have an increase, a slight increase of the membrane potential. Makes it easier to generate an action potential. Or the neurotransmitter may be inhibitory. It triggers IPSP, inhibitory postsynaptic potentials. These two we have discussed in details in a previous part, so I'll leave it out. You can see here a drop of the membrane potential. Examples for IPSP neurotransmitters, GABA or glycine. Examples for EPSP neurotransmitters, glutamate or acetylcholine. Now, why does it take longer in chemical synapses? Well, the neurotransmitter, after it has been released, it binds to receptors, and then it has to be taken back up into the presynaptic neuron, in the case of acetylcholine, for example. This is time-consuming. Now, electrical synapses do not need these steps. They are very fast. Here we see clusters of gap junctions, electric synapses. A gap junction is referred to as connection. Now, what is a connexon? I have a summary here for you, the notes. Connexon is composed of 6 connexin monomers. You can see every single one of these is a monomer, connexin. 6 connexin together form a connexon, a junction. I'm indicating here, this is a two-way transmission for small ions or molecules. This is one plasma membrane, and this is another. Both membranes are connected by gap junctions, connexons. See how well aligned they are, forming intercellular channels. They can be either closed or open. The gating mechanism can be either electrical or chemical. Now let's take a look at some examples for electrical synapses. Prime example is the heart as a syncytium. We have here the excitation system of the heart. It starts off with the sinoatrial node. I'll discuss this in details in a separate part. Just remember, this is the site where the first action potential is generated in the heart, the sinoatrial node, the SA node. Now, this signal needs to be transmitted almost instantaneously to millions of cells. This is where we need the electrical synapses. Remember always, instantaneous signal transmission requires electrical synapses. Between the cardiac muscle cells, we have intercalated discs. This is where we also have the gap junctions. You see, we have here one cell transmitting the signal to the other, and so on and so forth. Millions of cells will receive the signal almost instantaneously. Another example from the GI tract, gastrointestinal tract. Please remember the peristalsis requires the activity of millions of cells almost instantaneously. Here, action potentials generated one after the other. Millions of cells will receive the signal almost instantaneously. Another example of electric synapses is in the brain. We have millions of neurons that need to be synchronized, again, almost instantaneously. This is where we need electrical synapses. However, in the brain, we of course have also chemical synapses. Now, the electric synapses can occur, for instance, I have notes here for you, in the neocortex, hippocampus, thalamus, and so on and so forth. Interestingly, in the brain, we also have combined synapses. Here you see a chemical synapse, here an electrical synapse, and here a combination. This comes from a very beautiful review in Nature Neurosciences. I have here the reference. The summary of this excellent review: interactions between these two forms of interneuronal communication are required for normal brain development and function. Another example of electric synapses is in the retina, as it turns out. These two I will discuss in details in a separate part. The retina. In the retina, we have many different types of cells, the so-called retinal layer. And it turns out electric synapses play crucial roles there. I have the notes for you here based on this excellent Nature review. Just to show you here, we have the photoreceptors, bipolar cells, horizontal cells, and so on and so forth. Again, I will discuss this in a separate part, but here to show you from this Nature review, the gap junctions in the retinal layers. I have many different notes for you. Okay? It turns out the gap junctions are almost everywhere throughout the retinal layers. They do the coupling between rods and cones. They are also crucial for horizontal cells and for the ganglion cells, and so on and so forth. In the last 2 slides, I have extra notes for you, summarizing what I have explained. So I hope it was helpful.